Common blood pressure drug slows aging and extends lifespan. Even in older animals, while many people today live longer than previous generations, those extra years often come with chronic health issues. Scientists are now working to make those later decades healthier by finding ways to slow the biological aging process, especially after age 65. One promising strategy is replacing strict diets with targeted medications. Caloric restriction, cutting food intake without malnutrition, has been shown to extend lifespan in animals like mice, worms, and even monkeys. But for humans, this approach is hard to maintain and can cause side effects like dizziness, brittle bones, and hair loss. That's why researchers are hunting for drugs that can mimic the effects of calorie restriction without sacrificing meals. Why aging research matters. Aging is the root cause of many leading diseases, including heart disease, cancer, and dementia, because it gradually pushes cells into dysfunction. Slowing down aging could reduce the risk of multiple illnesses at once, rather than treating each disease separately. Public health experts emphasize that even modest delays in age-related decline could save billions in healthcare costs and dramatically improve quality of life. Traditionally, drug development focuses on single diseases, but the field of geroscience takes a broader view, targeting aging itself as the underlying issue. At the forefront of this field are caloric restriction mimetics, CRMs, drugs designed to activate the same metabolic pathways the body uses to survive during periods of low food intake. These pathways help clean up damaged proteins, optimize energy use, and strengthen the body's stress response. Challenges of caloric restriction. Classic caloric restriction involves eating 20 to 40% fewer calories for extended periods, which has produced impressive results in rodents. However, the diet is difficult for people to follow and may suppress the immune system. Instead, scientists are exploring ways to trigger the same biological responses through medication. Modern drug screening tools now allow researchers to search vast chemical libraries for compounds that make human cells behave as if they were experiencing a calorie deficit. This technique not only speeds up discovery but often highlights drugs that are already approved for other uses, potentially saving years in development. Rilmenidine, a surprising anti-aging contender. One unexpected candidate is rilmenidine, a blood pressure medication that's been safely used for decades. After being flagged by machine learning models, rilmenidine was tested in Caenorhabditis elegans, a tiny soil worm commonly used in aging research. The results were striking. The drug significantly extended lifespan, even when given to older worms. According to João Pedro Magalhães, a molecular biogerontologist at the University of Birmingham, for the first time we have been able to show in animals that rilmenidine can increase lifespan. The drug's long-standing safety record adds to its appeal. It's prescribed globally for hypertension, with mild side effects in some cases like drowsiness, insomnia, or heart palpitations. Magalhães and his team are now exploring other potential uses for the drug. How the drug works at the cellular level. Rilmenidine acts on imidazoline receptors, tiny structures on cell membranes that regulate metabolism. One specific receptor, called NISH1, proved essential for the drug's anti-aging effect in worms. When researchers deleted NISH1, the benefits disappeared. Reintroducing it brought them back. Further analysis showed that rilmenidine boosted autophagy, the cell's cleanup process, and increased resistance to heat stress, two signs of improved cellular health. Notably, the drug didn't affect development or fertility, suggesting it specifically targeted aging rather than growth or reproduction. Testing in mammals. While C. elegans shares many genes with humans, further testing in mammals is needed before moving to human trials. To that end, the Birmingham team gave rilmenidine to mice. They observed gene expression changes in liver and kidney tissues that mimicked the biological signature of caloric restriction. Blood markers of metabolism also shifted toward more youthful levels. Because rilmenidine is already approved for human use, early-phase trials could focus directly on markers like inflammation, insulin sensitivity, and muscle strength, skipping some early safety hurdles. Its oral delivery also makes it more convenient than drugs requiring injections or strict diets. What's next for rilmenidine and anti-aging research? Long-term studies are still needed to confirm the drug's safety and ensure that improved biomarkers actually lead to longer, healthier lives. But the momentum is growing. With a global aging population, the benefits of delaying aging, even modestly, are enormous, said Magalhães. As research progresses, regulatory bodies will need to adapt to drugs aimed at aging itself rather than specific diseases. Ethical questions about access and fairness will also come into play. Still, the idea of taking a simple daily pill instead of following a grueling diet is appealing and increasingly realistic. If rilmenidine continues to show promise, future generations may find that living healthily into their 80s and beyond becomes less of a lucky break and more of a scientific breakthrough. The full study appears in the journal Aging Cell.